Along with the process of meiosis comes another process called gametogenesis. We referred to that term gamete before as a type of sex cell, or a generic term for a sex cell, a sperm or an egg. And genesis means to make new. So gametogenesis would mean to make new sex cells. So it's the process of making gametes, which would be sperm and egg, in structures called gonads. And gonads are just a fancy word for the testes and ovaries, which is where the sperm and the egg are made in humans. So there are two processes to make gametes, since we have two different types of gametes to make. We have spermatogenesis to make sperm, and oogenesis to make egg. Now I know that sounds like a really weird word, but another fancy term for an egg is an oocyte or an ovum. So that's where that term comes from. Let's look at spermatogenesis first. Spermatogenesis uses the process of meiosis in order to make sperm. So we start off with our generic cell that has two N chromosomes. We have two copies of each type of chromosome. So in this case, it would be 46. The crossing over in this version isn't shown. So we go through meiosis 1 and meiosis 2 to go from one cell splitting and eventually into 4 cells with half the number of chromosomes as that original cell. So they only have N chromosomes, one copy of each type. So we went from 46 down to 23 chromosomes. And after these 4 cells mature within the testes, they're going to mature into sperm that can then fertilize an egg. We have what we consider a 1-to-4 ratio, our original cell to 4 daughter cells. In this case, we have 4 active cells that will develop into sperm that can fertilize an egg. In the process of oogenesis, it's essentially the same. You notice that we have 2 N chromosomes in the starting cell, the parent cell. We go through meiosis 1, we go through meiosis 2, we get down to N chromosomes, but if you notice, there's one distinct difference. We end up with 4 cells. Only one of them is going to mature into an egg. The other 3 cells develop into things called polar bodies. They're essentially helper cells for the egg. But in terms of females, they're only going to have that one cell making a mature egg out of the 4 cells that get made. So we essentially only have a 1-to-1 ratio of parent cells to active sex cells in the end. We still end up with N chromosomes in the end. So if we had 46 chromosomes to start in humans, that egg is only going to have 23. The nice thing is when the egg and the sperm meet, you'll see in a second that we restore our chromosome number. Remember, each of those processes of gametogenesis is going to happen in the gonads, so in the ovaries and testes. When the egg and sperm fertilize, so the sperm is going to fertilize the egg, the nucleus of the sperm, which has N chromosomes, in the case of humans 23, is going to combine with the nucleus of an egg, which again has N chromosomes, or 23. So we know then N plus N would give us 2N, so 23 and 23 would restore our diploid number of chromosomes to 46. So the nice thing is we now know that once we have that fertilized egg, which we now can call a zygote, that's a fancy name for a fertilized egg before it starts dividing, that zygote is going to have 46 chromosomes, or 2 copies of each type of chromosome. We restore that 2N number. So we have 2 copies of each type. One came from the sperm, one came from the egg. There are some errors that happen during meiosis as well. There are 2 main errors that we're really going to talk about, but one of them is really just non-disjunction. Disjunction would mean to separate, and then not. So it means that the chromosomes have not separated equally. So as we're going through these stages of division, we may end up with an extra or a missing chromosome in the gamete or in our end cells. Because as they're dividing, instead of separating evenly, they don't separate evenly. Maybe 2 will end up in one cell, and none will get dropped into another cell. So depending on which one you get, you may end up, after fertilization, having a normal cell with 23, one that had 24 chromosomes, an extra one, to give us a total 2N number of 47 instead of 46. If this were to happen with chromosome number 21 in humans, and you ended up with an extra chromosome 21, so you would have 3, you would have a child that would have Down syndrome. They have an extra chromosome number 21 for a total of 3 chromosomes. If we were to have non-disjunction where that extra chromosome dropped into a different cell, and now our cell that's getting fertilized missed a chromosome, we would have 23 plus 22. We're missing a chromosome for a total number of 45, one short of the 46. If this were to happen with our sex chromosomes, we could end up with an individual with something called Turner syndrome. They're missing one of the chromosomes.